we are going to discuss the macromolecules that make up the human body. So this is the last portion of our review of the chemical level of organization, or chemistry. You should have been introduced to these organic molecules in your BIO201 course or similar general biology class. The first of the organic molecules is going to be carbohydrates. These are hydrophilic organic molecules, and remember, organic compounds always contain carbon. Carbon compounds do not dissolve in water because they have covalent bonds, not ionic bonds. They have covalent bonds. The general formula of a carbohydrate is CH2O to the N. So for instance, glucose, one of the very common carbohydrates, N equals 6, so the formula is C6H12, there's twice as many hydrogens as carbons or oxygens, and O6. So C6H12O6. There's a 2-to-1 ratio of hydrogen to oxygen and hydrogen to carbon. Carbon compounds are a good source of energy, so carbohydrates are mainly used for energy in the body. Carbohydrates can be put together in a number of different ways. Monosaccharides are isomers. They're going to be, for instance, one glucose molecule. Disaccharides are dimers. There's going to be two monosaccharides. So a glucose-glucose, a glucose-fructose, or a glucose-galactose. All of those are options. And when we put those two things together, we call those sucrose, that's a glucose and a fructose, lactose, glucose plus galactose, and maltose, which is two glucose molecules. In addition, we can make even longer chains of carbohydrates that we call polysaccharides. Some examples of polysaccharides are glycogen, our storage form of glucose, cellulose that we find in plants, and starch that we find in foods like potatoes and bread. So here's our monosaccharides. Our hexose sugars are going to be the main ones we use for energy. Our pentose sugars are found in DNA and RNA, and we don't use those as often for energy. So we have glucose, fructose, and galactose, and then our sugar molecules in our DNA and RNA are going to be deoxyribose and ribose. So notice that our hexose sugars have 6 carbons, and our pentose sugars are going to be in a ring of 5 instead of that 6 ring. Our disaccharides are going to be a combination of 2 monosaccharides. So these are the most common ones: sucrose, maltose, and lactose. Our polysaccharides are big, long chains of those monosaccharide molecules. Inside of our bodies, we store glucose as glycogen, and this is the branched form that glycogen will take on when it's stored. So carbohydrates, the functions of carbohydrates: all digested carbohydrates can be converted to glucose and oxidized to make ATP. So the main function of carbohydrates is energy production or ATP production. However, we can also use carbohydrates for other functions. Conjugated carbohydrates are carbohydrates that are bound to a protein or lipid. A glycolipid, which is a carbohydrate bound to a lipid, can be found on the external surface of the cell membrane. The lipid portion hangs out in the plasma membrane, and the carbohydrate portion sticks up and sticks out into the extracellular fluid. Our glycoproteins are very similar, except the carbohydrate molecule is bound to a protein instead of a lipid. These are also found on the external surface of cell membranes, and they are found in the mucus of respiratory and digestive tract linings. Glycoproteins have a tendency to pull water towards them, and that's one of the features that makes mucus kind of wet and sticky and slimy. Proteoglycans are another form of carbohydrate and protein, and they're just arranged a little bit differently. And these are gels that hold cells and tissues together. So proteoglycans are generally found not attached to the external surface of a cell, but actually as an extracellular product. These help with joint lubrication, and proteoglycans are what contribute to the rubbery texture of cartilage. Basically works as a shock absorber. Lipids are hydrophobic or water-hating organic molecules. They also are composed of carbon, hydrogen, and oxygen, but they're put together in a different way. They are less oxidized, and therefore they have more calories per gram. So lipids are very, very rich in energy. There are 5 primary types in humans, and we'll primarily talk about 3 of those 5 types in BIO201. We'll discuss fatty acids. These are basically big, long chains of carbons with oxygens and hydrogens attached to them. So a bunch of carbons all in one big, long line. We can have saturated fats, which are going to be saturated with hydrogen molecules, and we can also have unsaturated fats, which contain 1 or more double bonds between carbon molecules, because those carbon molecules are bound with each other instead of with hydrogens. Triglycerides is the form of fat that we store. We basically take 3 fatty acid chains and attach them to a glycerol backbone, and we store them away as triglycerides. A phospholipid is one of the main components of our lipid bilayer, our cell membrane. So it is a phosphate molecule bound to a lipid. So the phosphate portion is water soluble, the lipid portion is hydrophobic, and that's what allows our cell membranes to get along with a watery extracellular fluid and the watery cytoplasm, but still have a hydrophobic layer in the middle. The last 2 are basically signaling molecules that are created from fats, and one of the ways we transport fats and another format that we use in the cell membrane. So steroid hormones, you've probably heard of these, things like testosterone, estrogen, thyroid hormone. And then cholesterol as well. Cholesterol is a building block for those steroid hormones. It's a ring form of fats. And the eicosanoids are going to be a signaling molecule as well. You may talk about those in 202 a little bit, but the first 3 are the primary ones that we'll be talking about in 201. So here is a fatty acid. This is actually a phospholipid, so we can see a number of different things about fatty acids in this molecule. The fatty acid portion is this portion down here, this portion that's kind of that golden yellow color. Notice that the this fatty acid is saturated. All of the carbons have 2 hydrogens on them. This fatty acid down here has a double bond, so it is unsaturated. So these are 2 different types of fatty acids, but they are both considered fatty acids, big long chains of carbons. Our glycerol backbone is right here, and we'll see this again when we look at a triglyceride, and we see the phosphorus containing head. So here's our phosphorus molecule right here. Got a little crazy with my drawing pen there. So that's where a phospholipid comes from. So here's our triglyceride. Once again, you see those fatty acid chains, and this time you see 3 of them, because a triglyceride has 3 fatty acid chains with 1 glycerol backbone. And so we basically take water out right here and form those triglycerides. So when we put 3 fatty acid chains on a glycerol molecule, we also end up with 3 water molecules. Because in our body most chemical reactions are surrounded by water, we oftentimes just omit this portion of the reaction because it just goes into the pool of water that surrounds everything else. So this is a dehydration synthesis reaction. We're synthesizing or building a new molecule, and by building that new molecule, we're taking out water, which dehydrates that molecule. The important functions of triglycerides are to provide protection, insulation, and energy when needed. So they protect our organs. Basically, our stored form of fat surrounds our organs. It also is found in the subcutaneous layer, which provides us with insulation, just like a sweatshirt or coat. And we can break down these triglycerides in order to use those fatty acid chains for ATP or energy production. So here's our phospholipid that we saw before, but now we're going to highlight our phosphate-containing head, our glycerol backbone, and we have 2 fatty acid chains that are connected. So this is a standard phospholipid molecule, and it's going to be an important part of our cell membrane. It makes up most of the molecules in our cell membrane. So we've got a polar tail and a polar head and a non-polar tail. Steroids, in a very simple form, are going to be a group of rings that are connected together. So this is our cholesterol backbone for all steroids, and then we can basically rearrange from here to get our different steroid hormones. Our second to last organic molecule is the protein molecule. Proteins are a polymer, or a big long chain, of amino acids. The combination of amino acids determines the structure and function. An amino acid is a carbon with 3 attachments. So we've got our carbon group right here in the middle. And then we also have an acid group and an amino group. That N is part of the amino group. Sometimes that acid group is also called a carboxy group. And then each amino acid is unique because of the R group that it contains. So all amino acids have the carbon in the middle, the acid group on one end, and the amine group on the other end, but they vary in terms of their R groups. So the amino group is the nitrogen with 2 hydrogens, the carboxy or acid group is that COOH, and the radical or R group is that last little attachment onto the carbon. There are 20 unique amino acids, and they differ in their R groups, and those R groups are what gives them their functional ability. So we combine these amino acids in different ways to make different proteins that have different properties. Proteins have the biggest variety of functions within our body. We have structural proteins like collagen and keratin that make up our ligaments and our fingernails and our hair. Proteins function in communication and regulation. So some will be hormones and some will be cell receptors that those hormones can bind to and cause an effect. Proteins also function in membrane transport. Channels and carriers that move things through the membrane are proteins. Proteins also help with biological catalysis, or being biological catalysts. You may have heard of a catalyst in chemistry. It helps lower the activation energy of a reaction and cause that reaction to happen. In our body, we call catalysts enzymes. So an enzyme is a biological catalyst. It helps with lowering the activation energy and causing a reaction to kind of giving it a push, causing it to happen. Proteins also deal with recognition and protection. So proteins are antigens, antibodies, and clotting proteins. So they help our blood clot so that we don't just keep bleeding and bleeding when we cut ourselves. And our antibodies and antigens both are involved in identifying self and identifying things that are invaders and getting rid of those invaders. Proteins also deal with movement. So our molecular motors, myosin and actin, are molecules that repeatedly change shape and cause our muscles to contract. And finally, proteins are involved in cell adhesion, sticking two different cells together. So there are 4 levels of organization of a protein. The primary structure of a protein is the chain of amino acids and how those are put together. The secondary structure of the protein is the folding and twisting due to hydrogen bonds. Hydrogen bonds can cause alpha helices to form, or they can cause beta sheets to form. The tertiary structure of a protein is the 3-dimensional shape caused by the alpha helices, the beta sheets, and other bonds that form. So things like sulfide bonds that form between different parts of the protein molecule. That's the 3-dimensional shape. So you can remember tertiary is 3, so 3-dimensional shape. And quaternary structure is when we have more than one protein that comes together into a larger molecule. An example of this is hemoglobin. There's 4 protein chains that come together to make the overall hemoglobin molecule. That's quaternary structure. So proteins have a unique 3-dimensional shape that's crucial to their function. That's called the conformation, basically what shape they are. The ability to reversibly change their conformation is a unique property of proteins. So they can change shape, but they always pop back to their beginning shape. So this is how cell membrane pores open and close. So you basically have a channel that goes through, and some channels can open and close and open and close. Denaturation is when the conformational change occurs, so that 3-dimensional shape changes. But that is not a normal change, and when that change happens, it can't get back to the 3-dimensional shape it is supposed to be. So it's a conformational change that causes a loss of function. This can be caused by extreme heat or pH. This is what happens to the proteins in an egg when you cook it. The egg white starts out clear, and as you cook it, those proteins are denatured and they turn white. Enzymes are biological catalysts in living cells. Enzymes involve substrates, which is the reactant in the chemical reaction. It's the substance an enzyme acts upon. And the way we name enzymes is we name them for the substrate they act on with an ase at the end. So for instance, amylase, the enzyme, digests starch, which is officially called amylose. So whenever you see a name that has ASE on the end, it's a hint that it's probably an enzyme, and it probably digests whatever's in the first part of that name. So enzymes lower the activation energy, and they help to get a reaction started. They facilitate molecular interaction. So they basically take 2 things and they put them close to each other so that they can interact with each other. So without an enzyme, we see a higher energy of activation, this right here. So it's higher. With an enzyme, we see a much smaller energy of activation required. So these are chemistry terms. Hopefully you've encountered them in your basic chemistry class, but this energy of activation is much smaller, so it's much more likely that this reaction will proceed from reactants to products with an enzyme present. So our substrates are going to come together and they're going to go into our active sites. Our substrates are kind of like keys that fit into a certain lock. So the shape of the substrates is specific to the active sites. So these form an enzyme substrate complex, and then we use energy to put them together. This is an example of a dehydration synthesis reaction. Water is coming out. So we basically are taking a hydrogen from one of these and an OH or hydroxyl molecule from the other. We're creating water. We're also creating a bond that binds these two, in this case, amino acids or substrates together to make a product. Notice that the enzyme here at the end is exactly the same as it was back here at the beginning. So one of the features of enzymes is that they are reusable. So it goes through this process and then it can start this whole process over again. So some enzyme characteristics. They are highly specific for substrates. They only accept one substrate in their active site. They are reusable because they are unchanged by the reactions. They're extremely efficient and fast. They're subject to cellular controls, which means that we can turn on or off an enzyme or make that enzyme go faster or slower. Enzymes are also temperature and pH dependent because temperature and pH can change the shape of the enzyme and alter its function. Enzymes will vary in optimum pH. The enzymes in our saliva work best at a fairly neutral pH, but enzymes in our stomach work best at a very acidic pH. And the temperature optimum for most human enzymes is going to be human body temperature, which is 37 degrees Celsius. Some things that are associated with enzymes that are important to understand are cofactors and coenzymes. Cofactors are non-protein partners like iron, copper, zinc, magnesium, or calcium that bind to an enzyme and change its shape. These are essential to its function. So many of the enzymes in our electron transport chain, which makes ATP, have to have iron or copper in them in order to function. If they don't have those, they can't function and they can't create ATP. Coenzymes are organic cofactors derived from vitamins, oftentimes B vitamins, and they work as electron shuttles. So you can think of coenzymes like an electron school bus. Basically, they're going to load up a bunch of electrons on their little school bus and drive them over to the electron transport chain. We're going to talk more about how these work later in the semester when we talk about ATP production. But coenzymes are electron school buses, and they come from B vitamins. So an example of one of these coenzymes is NAD, and it transfers electrons from glycolysis to aerobic respiration. So it brings those electrons from glycolysis into the electron transport chain. So a metabolic pathway is a chain of reactions, each catalyzed by an enzyme. In this pathway that you can see depicted below, A is the initial reactant, B and C are intermediates, and D is the product. Intermediates are basically just in-betweens as we go from our initial reactant to the product. The naming or the convention when we write out a metabolic pathway is that we have our reactant, we have our intermediates, we have our product, and in between we have the enzymes that catalyze each reaction. So these would be enzyme names that catalyze the reaction that they're written above. So we can regulate this pathway by changing the activity of any of those enzymes in that pathway or turning the pathway off or on altogether. Our final organic macromolecule is nucleotides. Nucleotides have three components: a nitrogenous base, a sugar, that's going to be one of those pentose sugars, and one or more phosphate groups. So physiologically important nucleotides: DNA carries the genetic code, RNA assists with protein synthesis, ATP is an energy-carrying molecule, adenosine triphosphate, and cyclic AMP, or CAMP, can activate metabolic pathways. We'll talk more about CAMP when we talk about cellular physiology in the next chapter. ATP is the principal energy storing molecule in the body. It holds energy in covalent bonds, and the second and third phosphate groups are bonded with very high energy covalent bonds. So these little squiggles means that there's lots of energy stored in these bonds. So when we break these bonds, we release lots of energy. The enzyme called an ATPase is going to hydrolyze this bond right here and release the energy from that bond. So it separates the molecule into an ADP, adenosine diphosphate, an inorganic phosphate, and lots of energy. Phosphorylation is when we add a phosphate group back on. So when we have ADP, we've broken this bond and we only have 2 phosphates, and we go to add a phosphate back on, we call that phosphorylation. And we continually replenish our ATP supply. If we were to stop producing ATP, all of the ATP in our body would be consumed within 60 seconds. So it's important that we are always producing ATP. Cyclic adenosine monophosphate, or CAMP, is formed by removal of both high-energy inorganic phosphates, and we then turn the remaining molecule into a circle. So that's where cyclic comes from. It's triggered when we have a hormone that binds to the cell surface, and it becomes the second messenger in the cell. And it runs around within the cell and activates different effects. So it can turn things on, it can turn things off, it can cause transcription and translation to occur, it can cause channels to open and close, it can do all kinds of things within the cell. Our nucleic acids are going to be the DNA and RNA. Hopefully you remember these from your introductory biology class. The DNA contains the genetic code, which involves and is especially involved in cell division, sexual reproduction, and protein synthesis. It regulates the cell's activity, and there are 1 million to 1 billion nucleotides in a DNA molecule. RNA, or ribonucleic acid, has a ribose sugar instead of a deoxyribose sugar and comes in 3 types. It comes in transfer RNA, messenger RNA, and ribosomal RNA. It generally is about 70 to 10,000 nucleotides long, depending on which type of molecule it is. And these 3 molecules together are going to guide protein synthesis, which is coded for by the DNA. So that messenger RNA brings the message from the DNA to the ribosomal RNA, and the transfer RNA brings the amino acids to that group, and then we can create proteins. So you should be able to compare and contrast the physical and functional differences between DNA and RNA. Remember, the bases are a little different, the sugars are a little different. One is a helical shape, one is a big long string. So that would be a good review for you to compare and contrast those 2 molecules. See you next time for our discussion of cells.